let's talk about phospholipids. Phospholipids are very important, very important molecules in the body. They are what we call amphipathic, and amphipathic is a fancy word for meaning that they're part polar and part nonpolar. Okay, amphipathic, part polar and part nonpolar. And so phospholipids become very important in mobilizing lipids around the body. Why is that? Why are phospholipids important in mobilizing lipids? Can we think of that connection? So we said earlier that water is polar, right? Meaning it had that partial charge. Lipids are non-polar, right? Very neutral, very charge neutral, very steady, stable charge. And so they did not interact very nicely. We also said that water doesn't move, or lipids do not move around the body easily because the body is mostly aqueous, right? Now, a molecule like a phospholipid provides an enclosed region. So because phospholipids can sort of interact with both, they're middle ground for allowing fats to move through the body. And we'll talk about how they do that. But basically there's a hydrophilic head. So this part of the molecule likes water, right? It likes to interact with water, it's polar. This part of the molecule is hydrophobic, meaning afraid of water, and this part is non-polar, so it doesn't like to interact with water. It's going to interact with like molecules, which are other lipids, or non-aqueous environments, okay? And let's look at some examples here of how this can be used. So phospholipids with water help to create unique structures. So this is called a phospholipid bilayer. We should all be familiar with what this structure is. Where is this found? Where? Around cells, perfect. So it's the cell membrane. We'll talk about that next week when we look at cell membranes. But this unique structure is where we have a cell wall, which is formed by these phospholipids. Their non-polar tails are sticking in, facing each other. Their polar heads are sticking out, facing the aqueous environment. And this can either be the inside of the cell, which has cytoplasm, or the outside of the cell, where there's that extracellular fluid. Likewise, there's a micelle. A micelle is similar structure, but it's smaller, and its main function is for moving around fats. So fats move through the system, through the blood vessels, through the body, wrapped up in the center of one of these structures. This is called a micelle. So micelles help to mobilize fats in the aqueous environment of the body. Very important structures. And another example is surfactant. We'll talk about surfactant, but surfactant helps with that surface tension in the lungs and helps preventing the lungs from collapsing. It's a very important substance that's secreted in utero. It helps to keep the lungs healthy in utero while they're developing. And we'll talk about that later on in the course, right? So these are phospholipids, very important molecules for mobilizing fats, very important in terms of the cell membrane, and we'll continue to look at their structure throughout the course. Any questions on that one? Okay, let's move on. Eicosanoids. Eicosanoids are sort of a unique category. These are modified fatty acids. They're not very common in the body, but their function is in intracellular communication. So they help with cell signaling, which is how two cells will communicate. They help in cells sending and receiving signals. Examples are prostaglandins, which help to communicate, again, cell signals, thromboxanes, which help with blood clotting, and other types of signaling molecules. They're derived from arachidonic acid. And what we have here is this ring, and then these two extended tails coming off here. Okay, not very interesting, not a whole lot about them. And again, these are probably some of the unique examples we'll find in the body, which are cell signaling molecules. The last group that we'll look at, of fats, is lipids. And lipids, is steroids, right? So steroids are basically derived from cholesterol. That's the unique property of this category. Examples are estrogen, testosterone, aldosterone. We all know these steroid hormones that are in the body. And these are unique in that they have this steroid ring. Okay, so the steroid ring, which usually has some type of functional group attached, is the unique principle of steroids. Alrighty, any questions about lipids? Any questions about lipids? Very important macromolecule. Okay, probably the take-home point I want you to understand with lipids is their functionality and how they are polar and non-polar in the case of phospholipids, and then non-polar in the case of the other lipids that we looked at. Let's move on to proteins. Proteins basically are polymers of amino acids. So amino acids are these subgroups that come together in these links like a chain to make proteins. So the building blocks of proteins, there can be lots of different variation here again. Most molecules in the bodies are proteins, hormones are proteins, signaling molecules are proteins, and so on and so forth. Insulin is a very important protein. And so lots of different variety in the way that these atoms are put together. Okay, we have different types of amino acids. We're not going to get into the exact type of amino acids, but let's look at the peptide bond. So this is similar to that condensation reaction we saw earlier, basically the formation of a covalent bond between the carboxyl group and the amino group on two amino acids. So these are two adjacent amino acids, can be X and Y. We have a carboxyl group on the end of one. It's going to interact with the amino group on the end of the other. We're going to have water coming off. Remember that condensation reaction we talked about? And then we're going to have that peptide bond being formed. This is now two links in a chain. Okay, that will continue to happen over and over and over again, and we'll get lots of different varieties of proteins based upon the different amino acids that are being formed in these links. Okay, now a peptide is generally a smaller protein, so anywhere from 2 to 50 amino acids, whereas a protein is a substantially larger structure, so more than 100 to, or more than 50, anywhere from 50 to 100 amino acids in that link. Let's look at some of the levels of structure with proteins. So levels of structure with proteins. So there are three sort of tiers in the formation or the structure of a protein. The primary structure, and we already looked at the primary structure. This is basically the peptide bonds from one amino acid to the other, right? Very simple. So we have two amino acids covalently bounded, and we get that peptide or that protein. The secondary structure of a protein is basically where we have these alpha helix sheets being formed. All right, so this is where these proteins now, or these peptides now, start shaping themselves into different structures. One example here is the alpha helix, meaning it's just going to start coiling on itself. Okay, this is the secondary structure within this protein. And we also see that similar phenomena that we saw earlier, the hydroxyl group and the amino group interacting. But the only thing here is that it's not two amino acids that are linking up. They're already linked. We have a secondary structure because this entire molecule is beginning to coil on itself. Another example is the beta-pleated sheet, which we see here. Again, same mechanism. These two groups are interacting with each other, but again, in a different mechanism that actually brings about the pleating of this molecule. Okay, so we'll see this with, these are just two examples of secondary structure, but basically that link that we formed before now starts to do different interesting things, and this is the secondary structure of this protein. The tertiary structure is where things get even more complicated. We have different secondary structures. So for example, this was a alpha helix, right? And so we could probably have a beta pleated sheet somewhere on here. And we have several different complex secondary structures now interacting with each other to bring about a tertiary structure, which is much more complex. And this is where we get the differences between proteins. So we have globular proteins that are probably more rounded. We have fibrous proteins that are probably more elongated. And this is where we get that customization or uniqueness among protein shapes. And then finally, the quaternary structure gets even more complex here. This example is hemoglobin. And we'll see different proteins that are actually linked into this one quaternary structure. The structure gets even more complicated at this stage, right? The quaternary structure. All right, any questions with proteins? The main take-home point with proteins is understanding the formation of that peptide bond, right? The interaction between the amino and the hydroxyl group, and then understanding the different levels of the protein structure is going to be the take-home points there. All right, let's talk about nucleic acids. Let's wrap up with this. So nucleic acids are basically the building blocks of our DNA and RNA. So this is our genetic material that's found inside of our cells. Now the nucleotide structure is basically phosphate groups, 5 carbohydrates, or 5 carbon ring, right? Which you can see here. So a phosphate group, this 5 carbohydrate ring, and then a base. Some type of base is going to be added on here. Now the base can be either purine, these are examples of purines, adenine and guanine, or the base can be a pyrimidine, right? So a purine or a pyrimidine can be the base on that group. And so the pyrimidines are cytosine, thymine, and uracil. And so between this mix, we get different nucleotides, either DNA or RNA. Now, DNA is deoxyribonucleic acid, whereas RNA is just ribonucleic acid, right? So either it can have just this deoxyribose, or it can be a ribose, which is this version here. Let's talk a little bit about energy transfer in our nucleotides. So basically adenosine, which is a major component of our energy transfer. So they can be monophosphate, diphosphate, or triphosphate molecules. And these are a way for us to store phosphate groups. So as you'll see, this is adenosine, which is the nucleotide. And we can add 1 phosphate, we can add 2 phosphates, we can add 3 phosphates. And so this is a molecule that allows us to store some energy. We'll see later on in this course that phosphate is one of the major energy currencies in the body. And being able to store it or remove it from adenosine is an important part of a lot of metabolic processes. The conversion of ATP to ADP is a very important process. That's some release of energy in the form of releasing one of those phosphates. Okay. There's also nicotinamide. We'll see this in the cell respiration when you look at the electron transport chain and how NAD and FAD are important electron carriers. So they help to store electrons, take them to another place, and then release them. We'll see how these 2 nucleotides are important in that function. So that's FAD, which is flavin, adenine,